To close off the day, we've made it almost through day one. We are closing off today's session with a pitch competition. We started this pitch competition last year. We partnered with Nucleate, which is, as you've heard, a student-led, non-profit organization that basically is aiming to create the next generation of bio-entrepreneurs. And last year we partnered, we got 9 fellows, they came and worked with Molecule for 6 months, and we've just really built this relationship, and so we decided to put the pitch competition back in. So we'll have Fabio and Komali, who are both part of Nucleate Germany, to sort of explain a little bit about Nucleate and to introduce the competition, and then we'll have our 4 contestants come in and pitch.
Okay. Hello, everyone. So, as Ella said, I'm Fabio. I'm the National Managing Director of Nucleate Germany. I'm here with Komali, who is the co-director of our Activator program. And today we're just going to give you a bit brief information about Nucleate, what we do, the activities we do globally, but also within Germany, and then hand it over to the early stage biotech startups that take place within our Activator program. So at Nucleate, what we kind of focused on is turning these academic projects into impactful ventures. So we take early stage projects, usually led by early career researchers, and give them the tools, the knowledge, and perhaps the capabilities to then commercialize their research and turn this into something actionable in the real world. As Ella said, we're a non-profit organization, and our goal is to empower the next generation of biotech leaders. So this can be anything from company formation, which can also come as part of the program, professional advancement for startups, for founders, and also in general building the whole next generation leaders of the biotech ecosystem. So this can be VCs, business development, and so on. And we're really big on community building. So Nucleate, as I'll come to, is a global organization. It's not just restricted to Germany. And we really utilize this global approach to kind of not only help the teams in each of the regions to expand beyond their region, but to bring everyone together to exchange ideas and to really push it forward. So briefly, our core principles. First and foremost, it's an educational mission. We try to lower the barrier of access, similar to DeSci, to biotech entrepreneurship. So introducing ideas to maybe PhD students, postdocs that haven't had them previously, then identifying these early stage startups, helping develop them, bringing together people for networking events to kind of spark ideas, spark conversations. As I said, so Nucleate started in 2018 in Boston. You can see here just how much it's grown over the years since then. So it's spread throughout the US, it's spread throughout Europe. We even have in Latin America and Asia now some chapters. This is just a region. In total, we have over 800 leaders in our leadership team, over 40 regions, and these 800 leaders are part of over 440 academic institutions. Our main program is the Activator, is this accelerator program, which on the next slide I'll tell you about, but we've developed for much further beyond this. So we do workshops, we have lecture series, obviously networking events, career development opportunities for leadership within Nucleate, but also for the Activator teams. But yes, the Activator, which the teams who will pitch now have just gone through, this is the main flagship program that Nucleate runs. It takes no equity, charges no fees. It's all focused on biotech, but it's agnostic to the type of biotech. There's therapeutics, diagnostics, green tech. But we start with a team formation. So we try to, a lot of the teams that apply are either PhDs, postdocs. They may not have a fleshed out business plan yet. So we try to pair them with business founders that can maybe help in this area. We provide hands-on workshops led by experts. So in IP, strategic fundraising, market risk and application. And these are all tailored in mind to the teams that we have running the program. And finally, we have a final pitch showcase, which for Germany is on Friday. And then this follows by the Global Summit where we try to bring together people from all of the different regions where Nucleate is. These are just some numbers to kind of show that the impact the activator has had. So these are globally. So since 2018, since the first activator was run, over 100 companies have been formed, over 400 million USD has been raised, close to 300 biotech jobs, and 1 acquisition of a company. And on the right-hand side, you can just see some of the bigger names that have come through the program and are now alumni. And as we keep going, the idea is this alumni, this community gets bigger, and it just keeps feeding into each other. I thought I hit this slide. So in terms of the 2025 cohort, so we have 200 venture teams. This is globally. You can see the breakdown in distribution here. So as I said, quite agnostic. I mean, there's a large proportion of therapeutics, but it covers a lot of biotech areas. Specifically in Germany, we had 9 finalist teams. You're going to hear 4 of them pitch today. And this is just quick for some of the events that we have upcoming in Germany. So as I mentioned, we have the final pitch showcase in Munich on Friday. So if any of you are in Munich, feel free to come down. But if you're based in Berlin and you're not going to make it, on the 23rd of September, we also have a Bio-Innovation Day. So this will feature startup bootcamp in the morning. We're actually open for startups to apply now to see and then we'll run through and see which ones we accept. And then in the afternoon, we'll have a public session where there's panels, keynotes, pitches, and networking. And this is, thank you, we're a non-profit organization, so our sponsors that kind of make all our activities possible. And then I'll hand over to Komali, who will introduce our teams.
Okay. All right, everyone. Let's get to the most exciting part, that is the pitch competition of Nucleate Germany activator teams. In the next few minutes, you will be hearing back-to-back pitches from 4 high-impact pitches from 4 teams who are researchers who are bringing their ideas, their science from lab to real-world ventures. So at the end of the presentations, you, the audience, will decide or will vote for your favorite. Hence, get ready with your phones and to choose your favorite. With that, first team up is Chandrashesh Ghosh, who will be presenting auxiliary therapeutics. Unfortunately, he couldn't join in person today, so he will be presenting it virtually. And he's building a next-gen therapeutic platform to revolutionize the infectious disease treatment. Okay, can we have CD presentation? Yeah.
Hi, good afternoon, everyone. My name is Chandrashesh Ghosh, and I would like to thank the organizers for giving me the opportunity to present Olexia Therapeutics to you. And so the global goal of the company is to transform toxic drugs to make them safer, cheaper, and better. And to do that, I will tell you about a problem that is plaguing human health right now, and that is fungal infections and mainly invasive fungal infections. So all immunocompromised patients suffering from cancer, HIV, or people who've received new transplants or ICU patients actually suffer from invasive fungal infections. Around 6.5 million is the number that we look at. And around 4 million people die due to invasive fungal diseases. And all of the patients that I told you above actually succumb to these fungal infections, around 20% of them. So it's a problem that needs to be solved. And one of the leading drugs against that is amphotericin B, which is in the list of essential medicines of WHO. And this can be used for treating infections caused by Cryptococcus, Aspergillus, Candida. These are all pathogenic fungi, and also a parasitic disease called leishmaniasis, which is affecting a lot of people in South America, parts of India, and also parts of Africa. Unfortunately, amphotericin B, although it's a great drug, is extremely toxic against liver as well as kidney. And it is absolutely not soluble in water, so you cannot take it like a pill, like orally. It needs to be formulated and given as an intravenous injection. And if you formulate it with lipids, for example, then the cost of using a standard 15-day treatment of amphotericin B lipid formulation is around 18,000 euros. And you can appreciate people in India, South America, and Africa would not be able to afford this sort of treatment. So we need to come up with a formulation of amphotericin B, which is water soluble, not toxic, and as well as affordable. And to do that, we have a new formulation, which can, in principle, it's like a platform technology of sugar-based gold nanoparticles, and we have conjugated amphotericin B to it with 2 to 3 very simple steps, and you get a 10 nanometer nanoparticle of amphotericin B conjugated to gold nanoparticles, and it is extremely soluble in water, till 10 mg per ml, to be honest. And if you look at it, if you leave amphotericin B in water, it falls down like this, and this is the formulation. You can see it's a clear see-through like amphotericin B in DMSO. And it's non-toxic, and we have shown this by a hemolytic assay. That means the red blood cells, if there is a toxic agent, it bursts and releases heme, as you can see here. Amphotericin B releases heme in 12.5 micrograms per ml, and this is the formulation at 64 micrograms per ml, which is much, much more therapeutic index. And you can see that the properties of the formulations actually become more effective, 2 to 4 times more effective. We've done tests against the pathogenic fungi that I described before, and we have looked at 50 clinical isolates from hospitals across the world as well. And we've seen that every time the formulation is 2 to 4 times more effective than the parent drug. Apart from that, some of these fungi can form these things called fungal biofilms, and amphotericin B is not so good at treating these fungal biofilms. However, the formulation is 2 to 4 times more effective than amphotericin B at treating these biofilms. And similarly, some of the fungi can form these vesicles inside your immune cells and survive within. For example, Cryptococcus, and amphotericin B, the parent drug is not able to do it so well, and the formulation is very good at it. It is also very effective against leishmaniasis, and the IC50, that is the concentration at which 50% of them are killed, is brought down from 1 microgram per ml to around 0.1 microgram per ml, which is very, very good. And in comparison to all the other formulations that are there in the market, we have a less toxic, very efficacious, and very, very cheap formulation, which is the next generation therapy. And one good thing about it, you can just lyophilize it, put it like in a powdered form, and probably use it orally as well, just by putting water into it and taking it as a pill. And we are trying to take this particular formulation forward for amphotericin B into a market which is around 16 billion strong. The lipid formulation Ambiazome alone actually generates around more than 500 million, and we want to tap into that market, which is going to grow at a CAGR of 6.5%, and also fueled by the aging population of the Western world, and they will rely on this kind of drugs. And of course, people back in India or in South America also would require these formulations a lot. And where we finished the laboratory models, and the animal models are ongoing at the moment. We finished the preclinical toxicity model, and I can tell you it's doing very, very well. Hence, I am out here to raise more funds. We are looking to do for 2 to 5 million euros to complete preclinical studies and then go into the next round of phase 1 clinical study. And we are willing to partner with any big pharma, and also are willing to be acquired by them at a given point in time, because the formulation is doing very well. And we also have ambitions of expanding this nanotechnology into other diseases to make their drugs soluble and less toxic. And I am going to be the CEO, and I'm generously advised by Professor Seeberger, who's the director of the Max Planck, where the work was initiated. I'm supported by Nucleate. Thanks, guys. Max Planck Foundation, NIH, and also we're looking for team members to join this team. And I'm very happy to have any questions that you might have. Unfortunately, I apologize that I couldn't be there in person for personal reasons, but I hope you guys have a successful conference, and I'm willing to hear from you. Thank you so much. Bye.
Negar, who will be presenting us with Exosphere. Negar is a medical engineer, and combining her technical expertise and her vision for early detection of cancer was where the Exosphere was built from. With that, I welcome Negar.
So, hello everyone, my name is Negar, and I'm very excited to be sharing with you how at Exosphere we are saving your lives by introducing early cancer diagnostics. You might be asking, why my life? Because in this room, out of every 2 people, 1 will be diagnosed with cancer at some point in their lives. And unfortunately, the diagnosis will be likely at a very late stage. And when you're diagnosed late, your chances of surviving this disease is significantly lower, by even up to 14 times. If you look at aggressive cancers like the pancreatic one, you can see that diagnosis at stage 4 has only 3% chances of surviving. And it is more scary to know that almost half of the patients are diagnosed at that late stage. It is crucial to diagnose the patients early on. But unfortunately, right now, there is no accurate and cost-effective solution that you can utilize as a screening method and to not wait for the symptoms to appear on the patient. But I'm not here to just get you scared today. I'm here to also give you hope. Because at Exosphere, we are building a solution to this. We have built a system that can target and identify the biomarkers of cancer inside blood. How it works is that you put the sample in a very small microfluidic kit, which is this big, and you insert the kit in a reader device, and you can get a signal readout, which is the indicator of the concentration of the target cancer biomarker. The innovation of this system is that it combines a low-cost test with a very high sensitivity. Even if you are at the very early stages of the disease and the tumor is only shedding a small amount of molecules into your blood, this system is able to identify those. Our business model consists of selling these kits and the reader to diagnostic labs and centers where this test will be then provided as a service to the patients. Long term, we would like to also have this reimbursed by insurances. There are several companies in the liquid biopsy market, but if we look at the 2 important factors, again, for early screening, what's important is cost and sensitivity. We can see that a lot of them have a sensitivity very low, even as low as 40%, and costs that are above $1,000 per test, up to even $4,000, which makes them not very suitable for screening. Exosphere is, oh my God, the colors are bad, but you can see it's the basic 2-sided matrix is the only solution that combines the high sensitivity with low cost. If we look at only one indication, for example, pancreatic cancer, we can see that the addressable market for this technology is very high, to billions of dollars. But what is important to keep in mind about this technology is that it is a platform technology, meaning with minor adjustments, we can adjust it for other indications of cancer, as well as infectious and cardiovascular diseases. So not only the target market is very large, but also the business is highly scalable. So far what has happened is that we have tested this system and our kits with several different biomarkers that we have listed here, and we have seen amazing results. And at the stage that we are right now, together with Germany's biggest diagnostic company, we are doing testing with patient blood samples and plasma samples to also show data with relevance to clinical samples. At this stage, we're also looking for angel investors to join us and help us with the financing of the round. Our team comes from several different backgrounds, from technical, biotechnology, biochemistry, but we all share a vision of saving patients' lives by bringing innovations to market. And last but not least, we are thankful to our advisors, many of which who come from the Nucleate program, who have been supporting us in different aspects along our journey. Thank you very much. I would be happy to chat with you after the pitches and looking forward to the conversations. Thank you.
Yeah. The next up is from Stefan, who will be representing Onsera. Stefan is currently a research group leader who is pioneering the translational oncology initiatives at LMU Munich. Today, he will be presenting the Onsera platform where it works on this early cancer diagnostics related. Yeah. Over to you.
Thank you. Thank you, Komali.
Thank you.
Together with Max. Yep. Okay, let's start. So we're presenting you Onsera, a collaborative project between Helmholtz Center Munich and the LMU. And it's about the usage of tumor avatars and structure-based AI for target discovery in solid cancer. This is Thomas, and he was diagnosed of metastatic liver cancer. And he was one case of 18 million cases each year, but it was a very personal case as I accompanied him and treated him during his disease course. And he had not the luck of having a targetable mutation, but he needed general therapy for his cancer and had severe side effects, and his tumor did not respond to it. So we had to change it to another general therapy. Again, severe side effects. Again, his tumor did not respond to it, and ultimately he died. His case is representative of the majority of solid cancer cases and highlights the urgent unmet need for better targets and treatments. There is hope on the horizon, as last year a study was published that showed efficacy of Enhertu, an antibody drug conjugate, even in low to ultra low abundance target patients. However, these new therapies like ADCs, bispecifics, and CAR T cells, they simply do not work so well in solid cancers. Why? Because we lack really solid targets. And why do we lack solid targets? Because the classical target discovery approaches fail in patient proximity, for low abundance targets, and the turnaround time. So, what are our solutions? First, patient-derived organoids. However, these do not represent so well the patients, as the success rate and take rate is often not so good. We developed in our lab new methods and new media formulations to increase the success rate. And from now on, we call them tumor avatars. As you can see here in this RNA-seq analysis, they cluster very well with the original tumor tissues.
Thanks. So, together with our AI approach, these technologies form a synergistic platform enabling a groundbreaking target discovery pipeline with unmatched advantages. So, okay, so this slide should go step by step. So at Helmholtz, we created a structure-based AI model, which has the beauty of not only identifying targets on the surface of proteins, but also aids in drug design. And this has the potential to lead to completely new patient therapies. So using our Beam AI, we call it, we in silico create multiple binders, and subsequently we validate them with our tumor avatars. So here you should see a video of an immunofluorescence tumor organoid, and we have internalized it with an E-cadherin antibody which diffuses through the gel, binds to the membrane, and even is internalized. On the right-hand side, we have the second technology, our AI system, which creates a higher proportion of high affinity binding compared to our non-structure competing systems. So while other competitors primarily rely on diffusion or sequence-based models, these non-structure-based models often struggle with identifying new target spaces, and they lack the direct patient relevance. On the other side, our organoid-based competitors, they are closer to the patient model. However, they lack the AI integration, which is needed to scale and automate the discovery. So with our approach, we combine the best of both worlds. We are close to the patient-derived models with our tumor avatars, and we use our structure-based AI. This leads to several key advantages for our partners. First of all, we have access to a new target space, and we have a reduced failure rate due to the higher clinical relevance. We have a faster validation, and we are in regulatory alignment with the FDA's roadmap plan to phase out animal tests. So this is where we stand today. So we tested both of the technologies separately, and as a next step, and with your help, we will validate the synergy of both technologies. We will file a patent for the AI and avatars, and we deepen our interaction with pharma. We organize follow-up financing, and we start a dual business model as CRO and co-development. Upon that, further scaling and exit. So Oncero is the team, and without our team, we would not stand here. So thanks to all of them, and thank you for your attention.
Thank you so much. The next up and the last one will be Henrik, who will be presenting Keynostics. He's the business mind behind the Keynostics team. And it's Keynostics is a diagnostic platform which is for vitamin diagnostics. So with that, I will hand over to Henrik.
Thank you. Hi, everyone. My name is Henrik from Keynostic, and what we are building is the novel diagnostic platform for personalized deficiency monitoring. Thank you. I want to present you later on our first use case, which is vitamin diagnostic. But before diving there into the deep end, I'll just gonna start with my hobby. I'm actually an ultra marathon runner. This basically means I run more than 40 kilometers by choice on the weekend through the mountains. What I ask myself there often is, do I actually have a healthy body? I guess this is a question which most people ask themselves, and if you eat healthy and you have a good lifestyle, most would say, I'm fine. But the question is it really true internally? And I will start with the topic of vitamin diagnostic. Now, on the left side, you see me during a run last year, 106 kilometers, 5,000 meters in altitude. And basically, I did a blood test before and after the run, and it showed me some constants, some markers went up, but also regarding the vitamin markers, we got a drop of more than 40% in that case, for example, for vitamin B12. What most people, on the other hand, take every day is medication. So also for medication, you have very specific indicators on micronutrient levels. So for example, a few 100 million people take metformin every day, and basically it has a clear case with B12 deficiencies. Now, you could go to a doctor repetitively, get your blood collected, send it to a lab, get the results, but it's very expensive and cumbersome. And even though there are 200 million vitamin tests performed each year, and this is only for Germany. And we are here to change that. What we are building is a novel diagnostic platform for personalized deficiency testing. We start the technology with building a lateral flow assay, which you might know from the corona test. We are basically using capillary blood, putting it on the pad, and having multi-stripes to determine multiple biomarkers at the same time. And to enable a quantitative readout, we are actually using a smartphone app to determine the bands. As this is a platform technology, it's not only for vitamins, but the next use case afterwards will be hormones as well. Now, for the patients in itself, it will be faster, easier, and do-at-home test. Now, let's look at the technology behind it. And as I'm the business guy, I keep it very simple. The black dot here shows low vitamins levels. But what does low vitamin levels actually mean? It means that the vitamin in blue here binds to a protein in gray, but you see a lot of non-binding sites. And this is where we come in. We are developing mimics. These are shown here in orange, and they fill up the open binding sites, and this is what we're detecting. So the very interesting part, number one, this is completely new diagnostic approach. No laboratory is going to do that. And secondly, it is extremely personalized because it shows your levels. Now, looking at the markets, we see extremely interesting markets. Of course, on the lifestyle markets, every vegetarian and vegan knows you should take B12. How many people do it in the correct dose? Nobody knows. Also, the whole topic of personalized supplementation. A lot of people take different kinds of supplements, but is it in the correct dosage or not? Also, nobody knows. Then at the medical applications, we already talked about the topic of drug treatment, but also the post-cancer treatment. For example, if you had pancreatic surgeries, your body does not absorb micronutrients as good as it was before. So there is already a medical need for testing repetitively. Now, where are we right now? We're currently doing a proof of concept and putting 2 markers on a rapid test. We are in the rounds for a pre-seed, finalizing that in September. And our goal is to have an MVP in 1.5 years. And that means we have a rapid test with 3 biomarkers and a quantitative readout. As already mentioned, we're doing a pre-seed, 990K. The interesting thing, we already have a technological lead investor driving that, and we are now rounding up the amount. And lastly, let's look at the team here. We have all the team from organic chemistry, biochemistry, engineering, and business already in there, but also a very good advisory board. And now, let's enjoy the evening. If you want to talk more, happy to, even if it's running or funding, I don't care. Happy to talk.
Yeah. That's all for all the pitches, and please give again a huge round of applause to all the teams who pitched it today. We will have a QR code now. Please, all of you scan your QR code and vote for your favorite team.
Everyone, you can vote, and then in 5 minutes' time, we will announce the winner. So everyone get voting. Okay, I have it on my laptop. Okay.
Yeah. We'll wait for the fill-in. So, do you want to refresh on us?
Yes.
Yeah. Tell everybody to sit. Should we tell everybody to sit and we'll announce? Should we tell everyone to sit and then we'll announce?
Yeah.
Hello? Oh. Okay, everyone sit down. We are going to announce the winners.
Okay, all right. Are we ready? Oh! And the winner is Negar from Exosphere. Applause. Correct.
Thank you so much.
Maybe you can say some words.
Oh, okay. I wasn't ready for that. Okay, yeah, thank you everyone. It was a pleasure to share our idea of Exosphere with you, and I'm very much looking forward to the networking. I'm happy to also get to know you and what you're interested in, and perhaps there will be also collaboration opportunities in the future. And thanks a lot to Nucleate for the amazing program that you provided us with. And yes, congrats also to all the other speakers and the startups, amazing pitches. Thank you so much.